Thank you for watching Biology News Live. I'm your host, Leisha Jhamnani. On today's news, we have a cell undergoing mitosis, and here's Biology University's Hudson Thomas to explain it to us. So Hudson, what is mitosis?
Thank you, Leisha. Mitosis is the division of the nucleus, which usually occurs in conjunction with cytokinesis, the division of cytoplasm, to produce two diploid daughter cells.
A diploid cell, of course, being one with two complete sets of chromosomes, one from each parent. Hudson, what was the cell doing before mitosis began?
Before mitosis, or the M phase, the cell was undergoing interphase, a time the cell spends growing and replicating its DNA in preparation for division. The vast majority of the cell cycle is spent in interphase.
I understand interphase has three portions, G1, G2, and S. During the G1 or gap1 phase, the cell grows and prepares for mitosis. The cell is beginning the process of division.
Yes, and next is the S phase, or the synthesis phase. During this phase, the cell makes identical copies of its chromosomes, a process called DNA replication.
Thank you, Hudson. The last portion of interphase is the G2 or gap2 phase. The G2 phase is when the cell will grow some more, finish preparations to divide, and create any remaining molecules needed for mitosis. The cell is now ready to divide.
And that's where cell is now, at the very beginning of the M phase, which is split into 5 stages. The first stage is prophase. During prophase, the DNA in the nucleus, formerly stored as chromatin, condenses into chromosomes, made of two sister chromatids joined at the centromere. The centrioles will move apart and start forming the mitotic spindle. After prophase is prometaphase, during which the nucleus dissolves and the spindle connects to the connector proteins.
After prometaphase, the cell will move into the third stage of mitosis, metaphase. During metaphase, the kinetochore proteins will move the chromosomes to the center of the cell, where they will line up to create the metaphase plate.
Now the cell moves into the fourth phase, anaphase. During anaphase, the spindle fibers shrink, pulling apart the sister chromatids. The chromatids move to opposite ends of the cell.
Next, the cell will begin telophase. Telophase involves the forming of a membrane-bound new nucleus and the transformation of the chromosomes to chromatin. The cleavage furrow also starts to form in animal cells and the cell plate in plant cells. And the cell starts to split in two. Telophase is the last stage of mitosis, but there's one more process that must be complete for the cell to divide. At the same time mitosis is occurring, the cell is also undergoing cytokinesis. While mitosis is the division of the nucleus, cytokinesis is the division of the cytoplasm.
This process looks different in different types of cells. Our cell is an animal cell, so a cleavage furrow is forming around it. This cleavage furrow, a ring of microfilaments, primarily actin and myosin, will slowly contract around the cell until it's completely split into two daughter cells. In plant cells, a cell plate starts forming these two future daughter cells and slowly grows out to both sides of the cell wall. Once the cell plate is fully developed, the cell will split into two. After the division, the cell plate becomes part of the cell wall.
So Hudson, as an expert on the topic, what do you think about mitosis? Is it a completely efficient process?
Mitosis is certainly an efficient process. Vast numbers of cells can be rapidly produced, and there's no conditions to be fulfilled, like finding a mate. Cells can simply enter G1 as soon as they're signaled to do so.
Of course, that is all true, but wouldn't the creation of identical daughter cells result in almost no genetic variation? What does this mean for a species that uses mitosis to reproduce?
Yes, lack of genetic variation will certainly make it very difficult for a species to adapt and evolve to any issues that could arise in their environment.
Thank you for that clarification, Hudson. Now the parent cell has completed division and resulted in the creation of two diploid daughter cells, each with its own full set of chromosomes. Mitosis is complete, and what's this? Another type of division? Looks like there's another cell undergoing meiosis?
Yes, Leisha, it's true. There is a cell that has started the process of meiosis. Now, meiosis is the process of one parent diploid cell splitting into two haploid daughter cells. Then, the two haploids divide again, and meiosis ends with a total of 4 haploid cells.
A haploid cell being one with only one set of chromosomes, half the full set we saw in diploids. Unlike mitosis, where we only witnessed one division, meiosis has a total of 2 divisions. A cell must go through Meiosis 1 and Meiosis 2 before it can be fully divided.
Meiosis, of course, begins with interphase, which looks much the same as it does in mitosis. After interphase, the cell begins meiosis 1. Prophase 1 is the first stage of this process. Firstly, tetrads will be formed from the joining of 2 homologous chromosomes, a process called crossing over, during which 2 chromosomes will actually swap parts of their DNA. The joining point of these 2 chromosomes is called the chiasma.
Metaphase 1 is very similar to the metaphase we witness in mitosis. The tetrads line up in the center of the cell, creating another metaphase plate.
Next is anaphase 1. The tetrads are pulled apart, forming 2 homologous chromosomes with new sets of DNA. The creation of new mixes of genes is called recombination, and the new chromosome forms are called recombinant DNA.
During telophase 1, the chromosomes have finished moving to opposite ends of the cell and are ready for cytokinesis.
Yes, the 2 haploid cells will be split. But unlike mitosis, these cells will be genetically different because of crossing over and independent assortment, or the random nature that chromosomes line up and are sorted.
Meiosis 2 refers to the second division in the meiosis process. This division will result in a total of 4 haploid cells, each genetically different, due to crossing over. These cells will only have one set of homologous chromosomes.
Prophase 2, the chromosomes could condense. In metaphase 2, the chromosomes form the metaphase plate. In anaphase 2, the sister chromosomes are pulled apart to opposite ends of the cell. In telophase 2 and cytokinesis, the cells divide once more.
Meiosis is now complete, as the diploid cell has divided into 4 haploid cells. Hudson, is there anything special you like about meiosis that maybe we didn't have with mitosis?
The thing about meiosis is that it allows for genetic diversity, which, as I mentioned earlier, makes adaptation and evolution much easier.
But unlike mitosis, it requires the cell to find a mate, which takes more time and energy than just reproducing through mitosis. I'm Leisha Jhamnani, here live with Hudson Thomas from Biology University. Thanks so much for being here, Hudson.
Thank you for having me, Leisha.
That's all for now. Thank you for watching Biology News Live.